Hello everyone, my name is Evelyn, and today I'm going to talk about the fascinating intersection between genetics, microbiology, and public health. Let's start with the experiment. Imagine we've identified a chemical called the unidirectional DNA replicator, which inhibits a Euro-bidirectional DNA replication process in E. coli bacteria. This chemical allows replication to occur only in one direction from the origin. What do you think would happen to E. coli under these conditions? I was very impressed with this question in my midterm exam because I only got less than half of the points. Remember this question, it is important and very relevant to today's topic. I will come back to this question later, but for now, let's explore the world of E. coli and how it relates to what we've learned in both my biology and global health courses. In our biology class, we've studied the structure and function of bacteria like E. coli. One of the most important aspects of the bacterial cell is their cell membrane, which is the semi-permeable barrier that surrounds the cell. This membrane is crucial for controlling what enters and exits the cell. It functions as a selective filter, allowing essential nutrients to enter while preventing harmful substances from entering. It also helps maintain the cell's internal environment by regulating the movement of ions and molecules. In the case of E. coli, this membrane plays a pivotal role in its ability to cause infection. When E. coli enters the human body, typically through contaminated food or water, it first has to survive the acidic environment of stomach. Once it passes into the intestines, however, it encounters a very different environment, a warm, nutrient-rich environment that supports bacterial growth. At this stage, E. coli uses specialized structures on its surface, like pili or fimbrium, to attach to the lining of the intestines. These structures act like little hooks, allowing the bacteria to adhere firmly to the intestinal walls, preventing them from being swept away by the flow of the digestive fluids. Once attached, E. coli can begin to release harmful substances known as toxins. The toxins can enter the cell lining the intestines, causing damage and disrupting their ability to absorb water and nutrients. And this leads to the inflammation, which is the body's natural response to the infection. And one of the hallmark symptoms of an E. coli infection is a bloody diarrhea. The disruption of cell function and the loss of fluids and electrolytes are the primary reason for the abnormal pain and the watery and bloody stew that people experience when infected with E. coli. Moreover, the toxins released by E. coli doesn't just affect the intestines, they can also enter the bloodstream and spread to other parts of the body. In severe cases, this can lead to more systematic complications, including HUS, a condition that causes kidney failure. The ability of E. coli to invade the intestinal walls, produce toxins, and spread throughout the body demonstrates the power of the bacterial cell membrane in facilitating infection. The membrane is not just a barrier, it is a crucial tool that bacteria use to interact with their environment and cause disease. Now let's talk about the experiment we've done in BIOL110 course, where we worked with bacterial transformation. This is where we introduced foreign DNA into E. coli cells using plasmids, which are small, circular DNA molecules. In one of our labs, we used the plasmid contain the antibiotic resistance gene. By introducing this plasmid into E. coli, we transformed them into resistant strains, and this process is crucial because it highlights how bacteria can adapt and evolve, just like how E. coli might gain resistance to antibiotics over time, making it harder to treat infections. We also studied the DNA replication process in this bacteria. In BIOL110 course, we learned that DNA replication is essential for cell division and survival. But if we interfere with this process, as in our exam question with the unidirectional DNA replicator, E. coli wouldn't be able to replicate properly. In fact, only one strand of DNA would replicate. This would potentially stop the bacteria from dividing altogether, and this could be a possible treatment method for controlling bacterial infections by halting their replication process. So how does all this connect to real-world public health? As one of my Global Health 280 course, we've studied the foodborne illnesses, and one of the key players in this field is the E. coli, especially the strain O157H7. This strain is known to cause severe symptoms like the bloody diarrhea, abnormal pain, and it typically lasts about 3 to 4 days. These infections often happen through the contaminated food or water, and they represent a major public health concern globally. Foodborne illnesses caused by E. coli, such as the one we've been discussing, are not only a medical issue, but also a significant economic burden, impacting healthcare costs and productivity. Understanding how E. coli replicates and interacts with human cells at the molecular level is a key to develop better treatment strategies and preventative measures. Environmental factors such as water contamination, hygiene, and farming practices contribute to the spread of these infections. In global health, we emphasize a connection between ecosystem health and human well-being, recognizing that outbreaks are often linked to the unsanitary practice and food contamination. Addressing these outbreaks require both biological insights and global health strategies to reduce the contamination risks. In conclusion, what we've learned about E. coli and its impact on human health ties directly to both molecular biology and public health. Whether it is understanding DNA replication or using genetic tools to manipulate bacteria, these concepts are fundamental in developing strategies to combat foodborne illnesses. At the end of my video, I'd like to take a moment to thank my instructor, Professor Lee, for his support, and my classmates for the collaborative learning environment. Thank you for attention, and I'm happy to answer any questions you may have.